This video is about cell signaling. It's some miscellaneous information, including some cell biology vocabulary that's not likely to be found in your textbook as far as definitions, as well as some advice and warnings about how to study this cell signaling material. Here are some terms that are commonly used in cell biology, yet surprisingly they're not really defined anywhere, specifically not in your textbook glossary, and they're actually really hard to find on Google as well. So let's start off at the top. So translocate, this means movement across a membrane. So specifically, if we had a membrane, then this would mean to move across either in or out. The next terms are influx and efflux. These are terms that refer to movement in or out of the cell. This implies that we're talking about ions, and it implies that it's passive transport, namely that the ion would be moving down its concentration gradient. Cellular location is an important one, so this is where a protein is found, for example, within the cell. And so this would include things like at the membrane, in the cytoplasm, or in the nucleus. Cleave means to break a covalent bond. It's kind of like a meat cleaver, where something would be broken in half. And basically this is implied as far as being used for a protein. That's one way that a protein might be cleaved. For example, the last 10 amino acids would be clipped off, where the peptide bond would be cut right at that location to remove 10 amino acids from the rest of the polypeptide chain. Recruits and stabilizes, these are some terms that we commonly use when we talk about proteins in a complex. We also use this as we talk about any sort of cofactors or other smaller molecules that are not amino acid based that also are part of a complex. And so recruits and stabilizes each are implying that these are stable ways that two proteins can interact with one another, and often this is due to activation or inactivation. Here's some more vocabulary terms, and so let's run through these. So conformational change is something we've been talking about since we started talking about protein shape. So conformational change is any sort of shape change of a protein, and in particular for cell signaling, active versus inactive forms are the key thing we're talking about. And typically for these multi-step procedures in cell signaling, then conformational changes are what are triggering one step to the next. Downstream targets refers to steps that are later in the pathway. So you'll see a lot of cases where we might have A to B to C, or A to B to D and E, something like that. And basically anything that is in this area, so B, C, D, and E are all downstream from A. And so if we say something's downstream, it doesn't necessarily mean it's the exact next step, but it's somewhere downstream or after. Intrinsic means the effects originate within, and one particular way you'll hear this is intrinsic enzymatic ability. Namely that we have some protein, and based on whatever domains it has, one of those might be an enzyme domain, for example, a kinase domain. And that means that that particular protein can do a particular enzymatic activity on its own, and it doesn't need a separate polypeptide, which is a standalone enzyme. Instead, it has the ability to do that itself. And basically this is just getting us to understand how the process occurs. So how many different components do you need? Do you need one protein and a separate enzyme, or can you just have one protein that can do both of these steps? And so intrinsic is a way to discuss that. Sometimes you'll hear about cytoplasmic tails or other sorts of tails. For cell biology, this is implied to be a discrete portion of a molecule, in particular a protein, and it's implied that it's towards the end, either the N terminus or the C terminus. Constitutively means constantly active, and in other words, it's impossible to inactivate a particular version of a protein. A lot of times we'll hear about this in terms of mutations. Crosstalk, this is when there are two signaling molecules which each can affect some of the same molecules. And this means that if we draw out something like this, let's say that here's a receptor protein GPCR and another one ECR, and each of them will activate, say, some sort of molecule called A. Then these actually, because they both are activating the same component, this could amplify the effect. So crosstalk can lead to some sort of enhancement or amplification of whatever the product is if both of those receptor signaling pathways are triggered at the same time. Or if one was an inhibitor and one was an activator, let's say that for protein B, GPCR is an inhibitor, but ECR is an activator, they could cancel each other out. And the main idea for this is that the reality is in a cell there are many different receptors, and so even if we understand one process, there may be some sort of larger scheme of what's happening within the cell, and so a cell is not a vacuum, and so multiple things may affect an ultimate output, whatever that is the effects of, say, B and A in this case. Okay, so I've got some advice and warnings about chapter 16. This is all about cell signaling, and so this is a pretty burly chapter, a lot of material. I think it's really rewarding and really exciting. This is one of the core parts of cell biology. And basically, though, it's pretty overwhelming initially because there's a lot of different steps, a lot of different components. So I want to give you some advice about how to study and what to study. So basically, for these signaling pathways, there's going to be a lot of individual parts or components. I like to think of this kind of like the game Mousetrap, where there's lots of these different little parts. So there's like this bathtub and the staircase and a cage, all that kind of stuff. And they're put together in a very specific order such that the whole process can work properly. So this is a kid's game. Maybe you have seen it, maybe you haven't, but the whole game is to put together these different components in the right order, and then set this marble off down the stairway and eventually capture a little plastic mouse using this little plastic cage. And so basically I think of cell signaling kind of like that. There's a whole series of events that are taking place. Each individual part has a role, and if any one of those parts is broken, it's likely going to derail the process. And so what we want to know is not just that the process is derailed, but we want to know exactly where is it derailed. And one of the reasons we want to know about this is because it helps us to understand the overall process and how to potentially come up with pharmaceutical or other biochemical remediation. So if this was a process in someone's body, that we might be able to mitigate it and have some healing from it. Okay, so anyway, for the cell signaling pathways, so for each of these individual components, I want you guys to know who it is. So basically, what's its name, what's its abbreviation. Sometimes things have more than one name, so knowing its alias is also really useful. I think it's almost always useful to know the full name as well as the abbreviation, because when you know the full name, then it can help you understand what it's doing. And it also helps you remember what the abbreviation is. If you only learn the abbreviations, you have to get them exactly correct, so there's no spelling errors with that. If you are learning the names, then it's okay if you have some minor spelling errors. So besides knowing who each component is, next know what it is. So specifically, what does it do in the scheme of this particular pathway? So is it a ligand? Is it a receptor? Is it an enzyme, secondary messenger, etc.? Third part, so how does it fit into the pathway? This is going to be a multi-step process. So does it fit in early or later? Is it an activator or repressor for something? Next, I'm going to jump down to number 5 about where the process is occurring. I mean the cellular location for this. So is this particular part of the process occurring, say, at the membrane, in the cytoplasm, at some organelle, or the nucleus. And so basically number 1 through 3, as well as number 5, these are all really zoomed in, looking at a very detailed mechanism. In other words, the very detailed steps of what's taking place. This fourth one here about when the process is occurring, this is more zoomed out, and this refers to what sort of cell type this is occurring in. For example, sometimes we'll compare signaling in a skeletal muscle cell versus a neuron. Other cases, we might compare the conditions under which it's happening. And so this, I think, is something that is really helpful to provide some context and help you to remember one signaling pathway versus another. It's also some of the things I'm going to give you for clues for kind of cueing into the process. So don't overlook this larger context about when the process is occurring. Okay, and some other advice about how this information is going to be used. So basically, the big theme about what I'm going to do is knock out something, some step, something, and then ask you what's going to happen next. So how far do we get down the pathway? What builds up? What doesn't build up, etc. And so sometimes I'll mess up two things. Sometimes I'll knock one thing out, and then another time, and then at the same time, I'll also provide a lot of a material, say something is constitutively active. And so you have to be able to figure out, so what's going to be the ultimate readout? Are we going to be able to get to the end of the pathway, or can we, do we still get stuck at something that's earlier on? And so that's why it's really important to know these steps of what's happening. And one more warning, as we get further into chapter 16 on cell signaling, some of the components get reused, and so they can be used differently depending on the circumstance. So don't get too attached to these individual components, and then do pay attention to when that happens, because those are places that you might trip up later. Okay, so here's another approach to memorizing the material. So basically, you'll see in your textbook that there are these very colorful figures that have all these protein blobs, and sometimes they have more details than you need. So what I suggest that you do is learn the abbreviated pathways, and here I have a summary of the GPCR signaling. And so this is a spin-off from movie 16.2, and I just showed this first part of it. And basically, this is my summary of what's going on. Some of the details that I keep track of are not what your book focuses on. For example, I don't keep track of exactly where the ligand binds. And in this case, I didn't keep track of which subunit of the G protein was activated. So, or that we followed with the adenylyl cyclase. So basically, having some sort of a drawing for this is really helpful. And I encourage you to come up with these and be able to basically spit these out on the spot. Probably write them down the second you get an assessment, whether it's a quiz or an exam. And then you also should be able to talk through the different steps here. And so this is a lot to write down if you're just going to give yourself like a quick kind of outline of what to remember. So you might come up with some more abbreviated ways than this, but at least initially this shows you step by step what's going on. And so I'm not going to run through these. I did that on a different video. However, this is one way you can see how these are the ways I would approach studying it, is have this visual here and then also have this step-by-step process. And again, I think one of the big things that you can learn from this is, so how the cell signaling works is that we have lots of individual parts, each of which has their own role. They work together in a very specific sequence, and if you screw up one of them, then pretty much everything afterwards gets screwed up. Unless, sometimes if you reintroduce something, then you can kind of rescue or fix the pathway and kind of pick up where it got left off. So you don't actually have to go through every single step in the pathway if you have a way to substitute for a particular part. And so these are some of the big things about our understanding about cell signaling, is that it's a very predictable fashion, and things are not just kind of happening willy-nilly. It's a very reproducible, very predictable set of events. With that said, it's pretty overwhelming that there are so many steps in the pathways, but I think it's really rewarding as you start to break them down into different parts. And while this is going to seem pretty disorienting at the beginning, it will show up with some themes as you move through this material. And there's basically two sorts of receptor signaling pathways we're going to refer to. And so my other advice for you is to make sure that you come up with a way to organize this material and kind of get yourself in the frame set that, okay, I'm working with a GPCR, that's where I've got this 7 membrane 7-pass transmembrane receptor. And so that's when I've got this sort of G protein going on. And so just kind of get yourself in that framework and then get yourself set up on a path to be able to solve the problems.